at the basic level, inflammation consists of this. There's some pathogenic invasion that happens, right? Some cellular injury, some vessel wall injury, some kind of injury that starts this process of inflammation. One of the first things that happens is the mast cells, pictured here, degranulate. Well, that's a funny name for explode, right? So the mast cells explode, and these granules, pictured here, those granules are histamine. And the histamine gets released from these granules, and that histamine has a role in pulling in parts of the immune response. So mast cell degranulation with release of histamine is one of the first things that happens. Another thing that happens is activation of the plasma system. And so that includes complement. Remember, complement is all those chemical messengers that complement the immune response, okay, and coordinate things. There's clotting that happens. That clotting cascade is part of this plasma system. And there's kinins that are released and other chemical mediators, okay? And then there's release of these cellular products. So the cellular products, such as the histamine that we talked about, but also other cellular products that we'll discuss, get released at the tissue level and help identify invaders, target them, or opsonize them so that they are found by the killer cells and destroyed. Okay? Now, the effects of all of these things like complement, clotting, and kinin are shown here. One of them is you get vasodilation. So the histamine, for one thing, causes vasodilation. That's why when somebody has an anaphylactic response, one of the first things that we do is give them volume, right? We give them wide open IV because they have this massive vasodilation that happens. That's in part due to responsive histamine, okay? The next thing that happens is we have this vascular permeability. So the vessels, as they dilate, the cells also spread apart more, and then we have leaking of fluid, right? And that fluid is transudate. That fluid just leaks out of the vessels, and that's where the swelling comes from, all right? The other thing that happens is we have this diapedesis of these white blood cells coming out into the tissues, and that forms pus, right? Once these white blood cells do their thing, they're spent, they die, and they form pus. Thrombosis happens in the vascular epithelium, and that thrombosis is part of the healing process as well. Because if the organism that is causing the infection, for instance, is an anticoagulant, then that would cause more bleeding to occur at that site and potentially loss of limb or loss of life. And the body has to sort of contain that infection. And so it uses clotting as one of the mechanisms, not just intravascularly, but also in the tissue area, to sort of arrest the loss of fluid and keep all of that stuff together, like the infection itself. And of course, when we have all these things, the vascular permeability, the cellular infiltration, and the thrombosis, that all adds up to pain. And the stimulation of the nerve endings results in pain. So what are the goals of inflammation? Well, if your body had to write down its goals, like we all do when we're starting a new job or whatever, it would say that it's going to use histamine, prostaglandins, and leukotrienes that will release from the injured tissues And that will increase the vascular permeability, causing vasodilation and getting those immune cells to the area. All right, that's one of their goals. Another one is to limit and control the inflammatory process. We don't want the inflammatory process affecting the brain if the only thing that's injured is I hit the end of my finger with a hammer, right? We don't want that massive total body effect of inflammation. We want it localized to that area. And that's another reason that we use the clotting mechanism to keep everything sort of sequestered. Okay? We want to prevent and limit infection. Again, the clotting cascade helps to do that. We want to interact with the components that are necessary from the adaptive immune system. We want to form antibodies to these invaders. And then, of course, we want to get the area healed again. And that takes time. So we have to first address the acute attack, and then we have to heal the area to recover from the injury. Here's another, and I happen to like this one the best. This is my favorite overview of the inflammatory response. And it has all the things that we talked about, but now it divides it into arterial, capillary, and venule, which is where all of these things are taking place. So the arteriolar thing that is not pictured before is this arteriolar constriction. So vasoconstriction that happens in the arteriole to limit blood loss. Okay, we have transudate that comes out here. We have the mast cell degranulation, and that attracts leukocytes to the area, which happens in the capillaries, right? These leukocytes come out and emigrate into the tissue area. There's platelet aggregation that happens to limit blood loss, okay? And in the case of an intrinsic injury, so some damage to the blood itself, say in a heart attack, for instance, that platelet aggregation is one of the processes that we try to overcome. We try to prevent that from happening. Okay? And then as we move into the venous side of things, we have infiltration by the macrophages. We have fibrin deposition, again, to limit or to encourage clotting at that site, to limit the extent of the inflammatory response. And that's just, I like that summary. I hope you do too, because we're going to refer to it every week from now until the end of the semester. So remember that monocytes are macrophages. That's it. Monocytes get produced in the bone marrow. They travel downstream. They go to wherever they are going to end up, and they become macrophages. That then is the reticuloendothelial system. We have a name for this. When those monocytes grow up and become macrophages, they go live in the and make up the reticuloendothelial system. Okay, that's monocytes, some mobile macrophages, and fixed tissue macrophages that live in all the tissues of our bodies. Well, maybe not all, but most. So we have Langerhans cells in the skin and subcutaneous tissue. We have lymph node macrophages that are in the lymph nodes. You can see some of them pictured here, right? So these are lymph nodes that have macrophages living in them. We have alveolar macrophages in the lungs, Kupffer cells in the liver. These are the Kupffer cells here, the purple ones. We have red pulp macrophages in the spleen, etc. So all of these macrophages then form the reticuloendothelial system, and they are a major component of the immune response. So complement is a big part of the immune system as well. But to be honest, I don't really want to spend a whole lot of time talking about it because it's very complex, and I'm not sure that it's all that clinically relevant. When it is clinically relevant, we're talking about a C9 deficiency. You can look that up, okay? And you can see that the C9 deficiency happens down here, but I don't know how that works. And let's just there's things we should concentrate on. This is not one of them. So I want you to know about complement. I want you to know that complement's job is to complement the immune response, okay, and the inflammatory response. And know that it can be activated by any number of methods. One, it can be activated through the classical pathway, or it can be activated through microbial antigens in what we call the alternate pathway. The classical complement pathway consists of these components. So opsonization is the presentation of the antigen, right? So opsonization is where the antigen is identified, and then phagocytosis happens as a result of that. Once the virus, for instance, or the bacterium has been opsonized, the phagocyte can attack it. The next thing that happens in this classical complement pathway is lysis of the cell. So we find some complement that identifies the cell membrane of the bacterium, for instance, and attacks that membrane complex. Agglutination means that we want to we want to handcuff all the bad guys and put them in a paddy wagon, right? Well, that's agglutination. We take all of the bad guys in an area and we stick them all together so that we can keep tabs on them. And that process of agglutination is accomplished through the complement pathway as well. All right, this one is really, really important. Neutralization of viruses. So one of the reasons that the COVID-19 vaccine worked early on better than it works now for preventing any disease is that the COVID-19 vaccine was more effective as a neutralizer. All right, so neutralization of the virus is by far the most important effect that we can have from from antibodies. There are some antibodies that are very good at neutralizing the antigens, and some of them are not. Some of them are good at opsonization, right? Some of them are good at opsonization, but not as good at neutralization. Chemotaxis is getting the right substances to the area. Activation of mast cells and basophils, that's another important role of complement. And then those inflammatory effects that we've been talking about, increasing blood flow, the capillary leakage of proteins, coagulation of interstitial fluid proteins, yada, yada, yada. Okay? That's the complement pathway, the classical complement pathway. The coagulation system is this process that we know. We have talked about this every day that we've been in nursing because we know that people get DVTs and we have to prophylax against that. We know that people get heart attacks and we have to treat that. And the way to do that is with anticoagulants, right? So we have all sorts of different anticoagulants that we can throw into the mix that essentially get us down to 10A, which is the final common denominator in the coagulation system. The whole coagulation system is part of the immune response. We're going to talk about coagulation later. Just throw this into the main group of immune response related things, and we'll cover it in more detail later. Kinin then is essentially the thing that initiates the clotting cascade, and it's really the thing that causes pain, right? So this prekallikrein and the kallikrein and bradykinin and all. Ooh, bradykinin. Remember that? Bradykinin. You've heard of bradykinin. If you know of people who get a cough on an ACE inhibitor, that's because of bradykinin, and that causes vasodilation and vascular permeability in the lungs, and that's what is responsible for the cough. So we've heard of some of these things before, but it's really the initiation of the clotting cascade at factor 12A that causes that is the kinin system. But wait, there's more. That's not all. The other important component of this inflammatory response, nitric oxide, we're going to talk about when we talk about inflammatory diseases such as coronary disease, such as adult respiratory distress syndrome. Nitric oxide plays a role. It's a mediator that comes from the vascular epithelial cells, and that nitric oxide causes vasomotion of the capillaries. So very, very important thing that was discovered by accident, as most things are in science, during some research about ACE inhibitors, of all things. So now we've talked about all these things. We've talked about complement, we've talked about coagulation components, we've talked about kinin, we've talked about nitric oxide. Wouldn't it be cool if we could just stop talking about all those things individually and give them a name as a group? Well, that's what we're going to do now. Because all of those things, when considered together, are the cytokines. So the cytokines include tumor necrosis factor, interleukins, prostaglandins, histamine, nitric oxide. All this stuff together we can just now refer to as the cytokines. All right? It's going to be very convenient. You're going to appreciate it. There are some cellular mediators of inflammation as well. And the cellular mediators are shown here. We talked last week about cell surface receptors. There are all sorts of cell surface receptors, many of which are mediators of inflammation, and they are listed here. So the pattern recognition receptors, or the PRRs, the PAMPs, you may have heard something about them before. I imagine that you've heard of toll-like receptors from time to time. Complement receptors, and then scavenger receptors. These are all cellular mediators of inflammation. Mast cells, we alluded to earlier, they are the things that contain these granules that are filled with histamine, and those bags or granules get released during the inflammatory process. So the activation that happens, or the mast cell activation happens from physical injury, chemical injury, immunologic processes, from these toll-like receptors that are picked up. And the chemical release can happen by degranulation, as we said, or through lipid-derived chemical mediators. So fat that gets picked up on the cell membrane, I almost said cell wall, but we know that's not a thing, on the cell membrane can activate release of this as well. When mast cells degranulate, they have a lot of contributions to the inflammatory process. So here are some of the cytokines and other chemicals and other messengers that are demonstrated. So when we have degranulation on this side, this is an immediate response, and then synthesis through that lipid metabolism that I described earlier engenders a long-term response. So the degranulation or the immediate response involves heparin, interleukins, tumor necrosis factor. It involves vascular effects. It affects neutrophils and eosinophils, all of which are attracted, or both of which I should say, are attracted to the site for one purpose, and that is phagocytosis. Phagocytosis. Okay? The synthesis side, which is the longer term response, also involves things like tumor necrosis factor and interleukins, but also growth factors, the prostaglandins, and the leukotrienes. All of these together are generally considered to be unwelcome. They are things that contribute to problems with long-term inflammatory processes. And those are things that we usually don't want. All right, so what are phagocytes? They are the cells that eat stuff, right? So there are three different types of phagocytes. We have neutrophils, We have monocytes and macrophages, and then we have eosinophils, which don't contribute a whole lot. They're a little bit phagocytic, but not tremendously. So neutrophils are polymorphonuclear neutrophils. What does that mean? Well, that means that if we draw a picture or we look at these under the microscope, we'll see multiple nuclei, right? So we have these dark nuclei here, and then we have the cytoplasm out here. But there are multiple polymorphonucleocytes, multiple nuclei. And so these are the primary white blood cells that show up at the scene. They're involved in the early inflammatory response, and they do things like ingest bacteria and dead cells and get rid of all the junk that's around in the early injury phase. They are short-lived, and they are part of the purulent exudate. The rest of the world knows that as pus, right? So they die, they turn into pus, they are no longer useful as pus. The second kind of phagocytes are monocytes that become macrophages. So remember that macrophages are just monocytes that leave home being the bloodstream, and they go through diapedesis and go into the tissues where they go to live on the streets, all right? They're street fighters, and they are an important part of the immune system, the innate immune system. And then again, eosinophils, mildly phagocytic. They are mostly part of parasitic infections and are released during allergic responses. So this is that process of diapedesis where the cell, the monocyte goes through this process of first margination, and then it goes through the vascular wall in a process called diapedesis. There's a chemotactic source, that is the inflammatory mediator, and the inflammatory markers are being secreted at this site. It attracts these macrophages, and they go there and begin the process of phagocytosis. Notice that monocytes are littler before they become activated macrophages. And when they become activated macrophages, they then become street fighters, and they have a much richer contribution to the inflammatory system than the monocyte does. How do these things get regulated? Well, again, it's from all these cytokines. Tumor necrosis factor, GM-CSF is granulocyte colony stimulating factor, and I forget the names of these things. GM is granulocyte monocyte colony stimulating factor. GCSF is granulocyte colony stimulating factor, but they are specific factors that are specific to this process, where the activated macrophages are processed in bone marrow and become monocytes and then macrophages. So what about the other granulocytes? Well, we talked about eosinophils, largely involved in allergic reactions. Basophils are very similar to mast cells in tissues. They have an important role in inflammation, including release of heparin, the anticoagulant, but also histamine, bradykinin, serotonin, things like that. Phagocytosis involves different steps. The first step is opsonization. First, the thing that's going to be engulfed has to be identified. That's the process of opsonization. That's up to the antigen-presenting cell to do, to recognize it as an antigen and present it to the cell, to the phagocyte for destruction. Engulfment is the next phase, where the phagocyte engulfs that thing. the phagosome is formed. That is an internal, like a lysosome. Like a lysosome contains digestive enzymes. A phagosome contains the ingested thing, whatever's been phagocytized. Then there's fusion with those lysosomal granules to break down the product, the phagocytized item, and then destruction of that target occurs. So, which cells are phagocytes? Again, remember that it's neutrophils, macrophages after their role as monocytes, right? After they get transported as monocytes to the tissue level, they go through the tissue, through the vessel wall, into the tissue, and become macrophages. And then the third phagocyte type is dendritic cells. And they really are one of the rock stars of the immune system. These little dendritic cells that are pictured here. Okay, so neutrophils shown here, 1, macrophages, 2, dendritic cells, 3. So we talked about the localized effects of inflammation, but what are the systemic manifestations of inflammation? Well, sometimes those interleukins and all those cytokines escape the local area and end up getting to the brain where they can cause fever, right? So cytokines go to the brain, stimulate fever by their direct action on the hypothalamus. Okay? Leukocytosis also happens. That's where the bone marrow is told to increase the circulating number of leukocytes. But hey, guess what? This is interesting. As nurses, when we give a dose of some steroid, we see the white count go up right away. Well, the bone marrow can't possibly increase production that fast that in an hour or 2 or whenever you get the next CBC, the white cell count is elevated already. That can't possibly happen. The bone marrow can't generate things that fast. So how does that happen? Well, one of the ways that leukocytes get increased so quickly is because most of them are not circulating in the blood. Most of them are sequestered, attached to blood vessel walls, right? So they're in the blood vessel like this, and here's the white blood cell that's stuck to this wall right here. It's attached. And when it's attached, it can't go anywhere until it is liberalized or released from that vessel wall. Now, what can do that? Well, those circulating things, those circulating cytokines can activate this white blood cell, release it from the wall so that it floats downstream and goes to look for something to kill. Okay? So that leukocytosis that happens is a result not only of the increased production from bone marrow, but also because the white blood cells that are sitting in reserve, waiting to get into the action, attached to the vessel walls, are suddenly released, and that's what increases the white count right away. Okay? As, for example, happens when we give a steroid injection. And then there's increased plasma protein synthesis. That's all these acute phase reactants like C-reactive protein, fibrinogen, all these other things that we see during the acute inflammatory response. And sometimes we measure these things. We measure C-reactive protein to see what sort of inflammatory condition a person's body is in. It's a good test, albeit not specific for what kind of inflammation, but it's a good test to tell us that there is inflammation present in the body. Chronic inflammation, on the other hand, is where this inflammatory response lasts for 2 weeks or longer, and it's usually due to an unsuccessful acute inflammatory response. So I have an example for you. I had a hordeolum, or a sty, in my eye. And it was supposed to go away after a week. I guess it didn't read the textbook and know that it was supposed to disappear right away. And so it was sticking around. So I went to the ophthalmologist. They did a horrible thing to me. They numbed up my eye, ostensibly numbed it up. It wasn't all that numb, I got to tell you. They pulled my eyelid inside out, cut it open, tried to release all the schmutz from in it. It was disgusting and horribly painful. But at that point, after that 2 weeks, we no longer were dealing with the acute inflammatory response that hopefully would have helped to resolve this problem. But now it's a chronic problem. And so now I'm still using eye drops, trying to get rid of it, but it's taking a lot longer. And that's because it is at the longer end of this, where it's now chronic inflammation. Okay? Now, there are some other things in addition to this unsuccessful initial inflammatory response that can cause chronic inflammation. One of them is high lipid content of microorganisms. So there are certain microorganisms that tend to cause a longer term chronic inflammatory response. There's also toxins that can do it. And if there's a large amount of particulate matter or physical irritants, say for instance you don't get the splinter out all the way, or you have some retained foreign body in there, those are responsible for chronic inflammatory conditions as well.